Hi, everyone. I'm Pang Fox, the Science Engagement Director for the CACNA1A Foundation, and I'm excited to be here today with Dr. Henry Colecraft, who has been a member of our Scientific Advisory Board since the organization was formed in 2020. The CACNA1A Foundation is thrilled to announce that it has awarded Dr. Colecraft a $75,000 research grant to further his work in developing a therapeutic strategy for CACNA1A related disorders, regardless of the type of variant that a person has. Dr. Colecraft is the John C. Dalton Professor and Associate Vice Chair in the Department of Physiology and Cellular Biophysics, and Professor in the Department of Molecular Pharmacology and Therapeutics at Columbia University Medical Center, or Columbia University Irving Medical Center. One of the focuses of Dr. Colecraft's lab is to understand how calcium ion channels work at the cellular level. So what helps regulate or control when and how these ion channels open and close, and how do genetic changes such as mutations in these ion channel genes lead to disease? I'd like to welcome Dr. Colecraft today. Thank you for being here to talk about the research that's happening in your lab and how it will impact our CACNA1A community.
Well, thank you, Pankong, for the introduction, and also thank you to you and the foundation for this grant, which will certainly benefit the research that's going on in our lab. Great.
All right, so before we discuss the specific research project that we are funding, we'd love to give our listeners a little bit of background on how you came to this work. You have been studying calcium ion channels for a long time and are widely regarded to be one of the leaders in this field. So could you briefly tell us about how you became interested in studying CACNA1A related disorders and why you joined the Foundation's Scientific Advisory Board right after it was formed?
Great. So yes, thank you. So I got interested in calcium channels actually during my postdoc at Johns Hopkins. This was all the way back in 1997. So I arrived at Johns Hopkins and I worked with a wonderful professor, David Yu, who introduced me to voltage-gated calcium channels, of which CACNA1A is one type. And so with respect to ion channels, nowadays my lab does a broad array of ion channels, but voltage-gated calcium channels really remain my first love as it relates to calcium channels, ion channels. So we have done, so there's always been a part of my research program since I started my lab in 2001 that's devoted to calcium channels. And we've studied them in different, answering different questions about how they work and how they function. And, you know, CACNA1A is one that we worked on sort of peripherally during those last 20 years or so. With respect to the work that we've done focused on CACNA1A, I think I can point to an email from my colleague, Wendy Chang, Dr. Wendy Chang, who's a neurologist at Columbia. And she sent me an email about a patient she had with epilepsy, some ataxia, and potential intellectual disability, and so forth, who had a mutation in CACNA1A. And she wanted to know whether we could sort of work, whether we're doing any work in that area, and whether we could look at that mutation and then build on from that. So based on that, we started a whole program. So now there's a whole research focus in the lab that's devoted to CACNA1A and CACNA1A mutations. And we've spent the last 5 years or so building the infrastructure and the tools to really be able to delve deeply into the problems associated with CACNA1A and try to come up with solutions. And general solutions are what we're looking at. And that's what I wrote my grant about. So with respect to the foundation, so once I started working, I met the president of the foundation, Lisa, as well as some other families. And after working with them for a couple of years, they intimated that they were going to start this foundation, which I thought was a wonderful idea. One of the things that's been most impressive for me, dealing with families from patients that suffer from CACNA1A mutations, is that it's really the passion that they've had to really push the envelope to try to get researchers to focus and collaborate to essentially drive forward the progress towards a cure. And so when they mentioned that they were putting together the foundation and asked whether I'd be on the scientific advisory board, I was extremely excited to join, and it's been a really great experience, and also been wonderful to meet other scientists that are interested in this problem and trying to come up with collaborations to move the science forward.
Well, we're certainly grateful that Dr. Chung reached out to you and brought you into this space, and obviously we're also appreciative of Dr. Chung as well, since she is leading our natural history study. So that's great. And kind of going off on what you were saying about meeting Lisa and meeting other families. You spoke at our scientific and family conference last summer in San Antonio, Texas, and you had the opportunity to meet even more individuals living with CACNA1A related disorders across the spectrum of the disease. So can you briefly kind of expand and talk a little more about how working with the foundation and meeting these families has influenced your research? How has this impacted how you think about developing treatments for the CACNA1A community?
Yeah. So as basic science researchers in an academic lab, we typically are focused on fundamental questions that we spend our time in the lab looking into test tubes and things to try to figure things out. And in a lot of cases, it's a really academic exercise, right? You're trying to advance knowledge and try to understand something you're curious about at a very fundamental level. So the conference that I went to for the CACNA1A Foundation was really impactful for me, because it's probably one of the only times that I've gone to a conference which has had the patients as well as their parents and families together with scientists, and certainly seeing the impact, the human impact of the disease and the mutations really had a profound effect on not just me, but one of my trainees whom I took to the meeting as well, and really brought home to the fact that what we're doing in the lab really, there are people out in the real world who are hopeful about the kinds of things that we're doing in the lab. So in terms of the impact that it had, I think it really put some fire under us to really focus in and really try to hone in on the things that we could do that would advance the translational aspects of what we're doing, right? And not just think about the basic part of it, but linking the basic to the translational and then driving that forward. I think it was one of the major things that came out of, and also the idea that speed is important as well. I mean, people are there are folks out there that really are looking forward to breakthroughs in science to come through, and so I think it had a really huge impact on us, and took our work from the abstract into reality. So.
Yeah, and I want to say too that I know that it had just as powerful an impact on the patients and the families there. I think it does help them, and it keeps their hopes alive too, knowing that there are brilliant researchers and clinicians out there that are working together to help improve their lives. So,
right.
So they are as grateful for you as you are for them.
Thank you. I mean, one of the things that I also got from that conference was meeting parents of newborn children, right? Who, of course, in their day-to-day lives have no reason to know what CACNA1A is, and all of a sudden they have a child and they don't meet the developmental goals, and so they go through the whole exome sequencing and find out they have this mutation. And so meeting some of those sometimes young parents and sort of talking to them really also was very profound, right? Because a lot of times, yes, when you have a newborn and something like this happens, I think it's potentially a very lonely thing. But I think that's where the CACNA1A Foundation's been a real godsend to people. And so So yeah, I think I was very impressed overall. First of all, with the foundation, with the parents, for their passion to show up and to try to learn and do what they can to advance things. And I think, yeah, had a huge impact on us scientists as well.
Thanks for those kind words, Dr. Colecraft. Okay, let's get into the science, right?
Yes.
All right. Could you summarize the research project that that we are funding, and explain how you envision this moving our community closer to effective treatments for CACNA1A related disorders?
Okay. So maybe first, I'll probably provide a little bit of context for looking at this problem from a scientist's point of view, right? So if we look into the ClinVar database, it's a database where pathogenic mutants of all types of proteins are present. If we look in the ClinVar database for CACNA1A, there are probably over 1,000 mutations that are listed in there, many of which are listed as variants of unknown significance because no work's been done on them. Some are listed as likely pathogenic and others as pathogenic. So there's a broad range of mutations that can potentially give rise to disease. The second issue is that the disease is not homogeneous. It's very broad spectrum. So you have, people with epilepsy, some with ataxia, some with a combination of all three. You have the hemiplegic migraines and so forth. And so there's a huge spectrum of phenotypes, huge spectrum of mutations. And so that potentially creates a problem when you trying to think about how to treat this disease. The other aspect of it is CACNA1A mutations, like many other genetic mutation diseases, is considered a rare disease, right? And as a rare disease, which is by definition occurs in less than 1 in 2,000 people, there are some practical considerations for the development of therapeutics for that. I think one issue for the big pharma industry, which is the main driver of developing pharmaceuticals, rare disease is a bit of a challenge because the economic models make it not viable for them to spend so much time doing the research to find potential treatments for just a disease that will affect just a relatively small number of people compared to the population at large. And so that is a challenge. So, and yet the big pharma companies are the ones who have the resources to really push and bring cures to these diseases as well. And so one of the ways that I've been thinking about the issue is to try to develop general solutions to a lot of these rare diseases so that they can be, bashed together, and in that way lower the energy barrier for a big pharma company, for example, to be interested in that. And so that's the background of how I've been thinking about the problem. So coming back to CACNA1A, all CACNA1A patients are what we call heterozygous, right? For in our bodies, all the proteins that we have, there are 2 copies of a gene that make it, or so what we refer to as alleles. And for essentially all the CACNA1A mutation patients, they have 1 normal allele, so they make a perfectly good wild-type protein. And then they have the mutant allele, which is one where during the process of development, a mutation occurred early on. And so there are 2 aspects of it. First of all, if you have just half of your CACNA1A, so you have a mutation, for example, that eliminates 1 allele completely, and you have half the CACNA1A present, that's called a haploinsufficiency. That still leads to disease, but I think you can readily appreciate the way to treat that disease is to enhance the amount of the wild-type protein so that it comes up to the level of having 2, right? And so that's one part of what we're trying to do. Since everybody has a wild-type allele, how can we enhance that wild-type allele to come up to to the normal type. And there are multiple ways you can think about doing this, right? So you need to make more of the protein. You can either make more of it, or you can let it hang around for a longer period so that its total density goes up. So our approach is to harness what's called the ubiquitin system. This is the cell's mechanism to trash proteins. So we can manipulate that in a way that it lets proteins hang around for a longer period. So if we strip ubiquitin from CACNA1A, we can make more, essentially double the amount present in the neuron. And so potentially for loss of function mutations where the channel isn't made, or nonsense mutations, loss of function mutations where the channel isn't functioning very well, or nonsense mutations where the channel isn't made at all, we could potentially just boost up the wild type one, and that would be sufficient. All right, for particularly the mutations that cause a gain of function, that is mutations which make the channel overactive or open too easily. It turns out that those ones are actually potentially cause a more severe phenotype at the patient level. So those channels have to be gotten rid of, and so that's the second arm of our approach. We'd like to simultaneously remove the mutant channel while we boost up the wild type channel, and we do that using a targeted approach, which will essentially only work on the CACNA1A that we've targeted, and so that's a general concept of it. And so with that, it doesn't matter really what your mutation is, because if it's gain of function mutation, we're really trying to get rid of it, right? And so getting rid of that will be one arm of the treatment. The second arm is to do the boosting of the wild type, which would benefit everyone. So that's what we were working on.
Thank you for that description. Yeah, I mean, I think that really nails this, the complexity of this disease, that having too little of the protein is detrimental, but having too much is also detrimental.
That's right.
So it's great that you found a way to harness the cell's technology and either keep a good protein around longer or get rid of a bad protein much sooner than, so that it's not around to cause those phenotypes that lead to the disease.
That's right.
Yeah, that's so wonderful. Okay, so last question here, Dr. Colecraft. What excites you the most about this project and continuing to work with the CACNA1A Foundation and community?
Yeah, I mean, I think what excites me most about this project is I mentioned that I started working on calcium channels back in 1997. And at that time, my perspective was very much focused on mechanistic understanding, really, really taking apart these channels and figuring out what makes them tick. And it's a very academic exercise. What excites me about this project is the potential to link what we've understood about this channel at a very basic level, and to actually link it to real life consequences for people, right? And I think if we are successful, this approach is potentially applicable to not just CACNA1A, but all ion channels, and indeed all other genetic mutations as well. And so I think there's a test ground for us, in that if we're successful at doing this, we can really potentially blow up and help many other diseases as well. Yeah. And I'm looking to continue my partnership with the CACNA1A Foundation. I think it's been wonderful working with you, Lisa, Sunita, and the other mothers in the and families in the organization. I think one of the things that is brought to my program is collaborations with other scientists whom I wouldn't have known otherwise, and that's been a very beneficial thing, both for the science as well as for us scientifically as well.
Yeah, that's great to hear. I mean, part of our mission is building this collaborative network between patients, families, researchers, and clinicians. And so we're really grateful to have you on our team and and continue to collaborate with us and other scientists in our network as well.
Great.
All right. So thank you, Dr. Colecraft, for your dedication to the foundation, being one of our scientific advisory board members and also carrying out the much needed research to develop treatments for the CACNA1A community. We are certainly excited and can't wait to see the results of this research project.
Great. And I'd like to just end by saying thank you to the foundation and to the patients out there for all the people who contribute to the foundation. We'll certainly be good stewards of this grant and really sow it into trying to develop the therapies that's so much needed in the real world. So thank you.